Next, we have Dr. Uh, Dr. Gulam Hussain is a pharmacologist. He's a pharmacologist with 14 years of experience in pharmacology and toxicological evaluation of traditional and herbal medicine. He's a scientist, research officer pharmacology, scientist 3 at the National Research Institute of Unani Medicine for Skin Disorders at Hyderabad, which is an institute under the CCRU, Ministry of Ayush. He has previously served as a lecturer in pharmacology and toxicology at NIPER Raebareli and as a chemical safety and regulatory professional and evaluator Gurugram. He earned his M.Pharm and PhD in pharmacology from Banaras Hindu University after seeking AIR 95 in GATE and University first rank in B.Pharm. He brings so as you can see, he brings himself with him an overall experience uh, in research, industry, and academics. And today he's here to present his talk on pharmacological evaluation of traditional Unani medicine, challenges and way forward.
Thank you, madam, for introduction. Respected chairs, dignitaries of the dais, researchers, and dear fellow pharmacologists. Today, I will discuss about pharmacological evaluation of traditional Unani medicine in particular. What are the challenges and way forward? First of all, I would like to thank my Director General, Dr. N. Zahir Ahmed Saab, who has allowed me to participate and deliver this presentation over here, and Professor Anil sir for giving me opportunity to be part of this IPS conference. So, as the tone is already set by our AD, Dr. Ghazala Javed Madam, that traditional medicines being a part of a culture all around the world, WHO has notified that almost all countries have one or other form of traditional medicine in place in their healthcare system, which are basically holistic approaches, and traditional medicine emphasizes nature based remedies and holistic personalized approaches to restore balance of mind, body, and environment. So, as opposite to conventional allopathic medicine, uh, all traditional medicine focuses on a balance between mind, body, and environment to maintain health. It is believed that herbal drugs are safer than synthetic drugs, but there is a rider. People generally think that they are being derived from natural origin, natural sources, they are safe. But this safety can only be ascertained while we systematically evaluate these traditional medicine and practices being used throughout the world. So we have to adapt scientifically valid methodology, and we have to show the world that these practices or these medicine, be it a herbal or any practice just like yoga or acupuncture or some massage therapy or panchkarma therapy being used in Ayurveda, these all encompasses some uh, traditional cultural roots, but we have to prove them scientifically, clinically, as well as preclinically that these systems work. So this I will skip because Madam has already elaborated a lot on the traditional medicines recognized by Government of India. I particularly discuss related to traditional Unani medicine, but same concept can be applied to Ayurveda and other Chinese traditional medicine as well. So what now I will be discussing some basic points which we as a pharmacologist, as a bench worker, we uh, are exposed to. So these particular topics I'm going to cover in this presentation, which are related to test formulation, their dosage regimen, drug administration issues, pharmacokinetic and bioanalytical challenges, ethics, particularly very important while we are designing clinical trials, and how to design trials of traditional medicine which are having multifaceted intervention and drug interaction aspects. So coming to test formulations, so when we consider test formulation in modern medicine, usually oral solid dosage form, which are tablet or capsule, are considered best as of now because of ease of administration, long shelf life, and a high patient compliance. But we are having so many formulations in traditional medicines which are now being upgraded to equivalent to tablet and capsule forms. There are some unstandardized, we can say, dosage forms which are in the form of semi-solid dosage form, some liquids, which are some powders like churnas in Ayurveda, or safuf in our Unani system of medicine, which are bulky in nature. So test formulation has to be standardized. There are batch to batch variation as well, because many of the traditional medicine, if we talk about pharmacotherapy point of view, they are herbal products or derived from herbal ingredients. So when we collect from herbal or mineral origin ingredients, there might be source variation, there might be seasonal variation, there might be a variation in the cultivar or species of the particular herb or uh, animal origin drug. So that may lead to batch variation. And our practices of processing that raw material are also not standardized earlier. But now, since the advent of good manufacturing practices and adoption of these manufacturing practices by Unani pharmaceutical industry or Ayush industry as a whole, these practices are being standardized, and there are minimal batch to batch variation in these formulation. Similarly, quality control, as I discussed, is a big issue for the raw material and finished product as well. So our Unani Pharmacopoeia of India or Ayurvedic Pharmacopoeia of India or Siddha Pharmacopoeia or Homeopathic Pharmacopoeia, now they have given stringent quality control measures in the form of monograph, which we have to comply. The manufacturer has to comply, and they have to do complete stability shelf life studies, and they have to show that under this given circumstances, in these storage condition, that product should be stable enough, and patients safety cannot be compromised. Patient compliance is a big issue, while dosage form is not in the standardized form, as I said, oral solid dosage form. Sometimes we ask patient, still we have some formulation which we ask patient to soak in the water and take as a form of decoction after boiling or cold infusion, just after straining the raw herbal ingredients. But these formulation have poor patient compliance as well as lack of uh, equality all the time, because once we ask a patient that you have to dissolve this pouch in 1 glass of water and boil for 2 minutes, somebody will boil for 2 minutes, someone will boil for 5 minutes and heat. So these things has to be standardized. So instead of these, nowadays we are coming up with the SOPs to prepare the particular extract in the form of decoction or infusion. So that is the way forward, that instead of asking patients to do this process, we have to give patient a better dosage form which is consistent in nature. Coming to dosage or dose, so dose regimen is the biggest issue in the traditional medicine, because an ancient text, be it any traditional medicine, it is described vividly by varying number of doses, dosage regimen. Somewhere you will find that 1 gram daily it is written. Sometimes you will find some 1 gram twice a day. Even in older texts you will not find grams, but instead you will find older metric system or empirical weighing measures. So that is the big issue. So it has to be standardized, and Pharmacopoeia Commission of Indian Medicine and Homeopathy has taken this issue forward, and they have converted whatever written in the classical text into metric system, and now they are uh, uh, standardize the dose. So ambiguity should be avoided, and whenever there is a doubt, you have to check the original classical text to see the description, detailed description. But as far as a pharmacologist is concerned, large doses poses always challenge for uh, difficult for administration. So we have to see how we have to administer large doses to animals. Sometimes we have doses as good as 40 ml, 60 ml per kg for human. So while we are converting those doses to animal equivalent based on body surface area conversion, it is coming very high dose which we cannot administer to rodent. So we have to see alternate approaches, whether we have to concentrate those formulation or we have to make some extract sort of thing, so that little bit uh, 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 knowledge you have to apply to standardize the thing. Then, as I discussed, seasonal and geographical variation are always there. Constituents in the plant vary depending upon their geological geographical condition. One plant grown in Himalayas will having a different quantity of the constituent. If we take curcumin content which from haldi, which is grown in the southern part of the India, it will be very high compared to northern growth haldi. So that has to be taken care of. That's why now pharmacopoeia and all agencies are emphasizing that you have to give passport data as well. Passport data means which soil condition, which agricultural practices has been adopted while cultivating these medicinal plants. That has to be standardized. And those is big issue. Pharmacotherapy is fine, but when it comes to procedure based therapies, like I said, acupuncture or cupping therapy or leaching therapy, so then those has to be decided. So what we can do, we have to fix the frequency of applying these therapy for how long. So for that task, our council has taken a very good step to publish standard treatment guidelines for various regimenal therapy and cupping and leaching. Already our guidelines are available, as madam has discussed. These are freely available at ccrum.res.in, so everybody can access them, especially for medical practitioner. Drug administration, already discussed that it is a big issue, since majority of drugs are raw ingredients, just processed somewhat like grinding and sieving, and then make into the form of tablet or capsule or just as such as a powder. So they are it is quite obvious that plant part are not directly water soluble. So you will not get a fine solution like normal saline to be administered via intraperitoneal or oral route. So what we get is mostly is a suspension which we have to administer through oral gavage to the rodents, or we have to make a thick paste and then we have to apply a particular solvent, usually water, to make to add a suspending agent, and then we have to administer into the rats. But sometime what happens with, especially with semi-solid dosage form, like we can see on downside, it is difficult to get syringeability, because if we are administering in the mouse, so hardly we can go up to 18 or 20 gauge uh, gavage. So sometimes they are not we are not able to syringe them. Then we have to see alternate option, whether we have to go for extraction or we have to even milling sort of thing. Then why how to do cell based experiment, because for testing cytotoxicity or some cell based experiment, and we are moving from apart from animal testing, we have to see the alternative testing procedures. So we need to treat cell lines uh, with our formulation. So as such, formulation cannot be treated, but we have to prepare different type of extract, and we have to use ethanol, methanol, or hydroalcoholic extract sort of thing. Then we can fair enough we can do the treatment with the cell culture experiment as well. So these are the challenges. Now coming to the pharmacokinetic and bioanalytical challenges. So as you can see that even one formulation can contain 5, 10 herbal ingredients. Sometimes herbal ingredients mixed with some mineral, and sometimes complex of both herbal and mineral. So that time, in one plant there might be some 8, 10 active ingredient. There might be thousands of molecule, but active ingredients, let us speak. We have to measure those in kinetic experiment. We have to measure some drug. Suppose we are doing kinetic of atorvastatin, we can measure atorvastatin using LCMS or something. But in plants, what you will measure? At least representative biomarker or analytical marker, we can say, it may be responsible for biological effect. That is fair enough. If at all it is not responsible for biological product, but it should reflect in the blood stream, then only we can say that it is bioavailable or not. So it is the most challenging aspect of the herbal complexes or herbal formulations, that we have to develop method for their bioanalytical analysis. Then we have to validate method. Due to advent of new technologies like HRMS or metabolomics sort of approaches, we can easily, not easily, but at least we can characterize some marker, analytical marker, to have some kinetic data of herbal formulation. So people are studying these kinetic data, and now so many work has been published from our Unani Council as well. Now coming to some clinical aspect, what we challenge, as madam has already discussed some clinical aspects, so I will quickly go through. First and foremost is we have to take the consent of participant. It should be informed consent. Then we have to see the risk benefit analysis of our traditional formulation, whether our traditional formulation has to be given for that particular condition or not, because there are so many condition for which we cannot give our traditional medicine, being these are some infective disorder, and they have a notified disorder basically, and there is a standard treatment guideline issued by the health authorities. So every pathological condition you cannot intervene. Then how to design a trial and how to take control arm, because when we talk about clinical studies, always people what first come into the mind is randomized control clinical trial. So that is the best in the power and validity. With traditional medicine we cannot always design randomized control trial because of several reasons, because we cannot match the placebo, we cannot have always a control drug from allopathic side, because we have to again have one investigator from the modern medicine. So we have to on case basis we have to decide what type of the trial will be feasible for that particular formulation or that particular therapy, because for therapy as well, non pharmacological intervention as well, we have to prove the uh, uh, clinical efficacy. And as already discussed by madam, that ICMR has come with the addendum to national ethical guideline for clinical trial involving human participant, which was published in 2017. So addendum came in March 2025, and ICMR reiterate that whenever whenever evaluating any trial protocol, there should be at least two expert, one from traditional medicine, preferably outside of the institute, and one from inside of the institute. Rest of the ethical committee member will always be there. And now, since the notification of new drug and clinical trial rules, you all are aware, 2019, our ethics committees which are doing clinical trial on traditional medicine, these committees are also being accredited by Department of Health Research, Ministry of Health, Government of India. So now this is a big step to increase the reliability and ethics aspect in the clinical study of traditional medicine. And one more important is ASU GCP. Uh, ASU stand for Ayurveda, Siddha, and Unani drugs. And Government of India Ministry, that type Department of Ayush under Ministry of Health and Family Welfare in 2013. They have in 2013 given good clinical practices guideline. So you may access them through Ministry of Ayush website, and that has beautifully given what ethical aspect you have to take care. So here, when we are coming to trial design, basically, so when we are designing the trial, we have to see some aspect. Not only we have to go directly by modern medicine aspect, like diagnostic criteria, inclusion and exclusion criteria. We have to see our system criteria, classification of diseases criteria. Just like in Unani, we have four type of temperament of the people. We can whole population can be divided four subtypes. So they have different phenotypic characteristic, they have different genotypic characteristic, so obviously they will respond to treatment differently. So they will not it is not always true that as per our traditional uh, concept, that all people will not respond therapy equally. Similarly, in Ayurveda, we have three types of prakritis, like Vata, Pitta, and Kapha. So it is considered that we can divide all population into three subtypes or three subgenomic phenotypic as well as genotypic characteristic. So that aspect has to be taken care when outcomes we are assigning, we are making protocol, So outcomes, usually what we do, suppose a hypertension trial, so we will see systolic BP should go down this much, and diastolic BP should go down this much. So that will be our assessment criteria. But our traditional approach should also be there, that how to we observe pulses, nubs में कितना changes है, so that concept should also be there. And then actual integration is possible. If we are recruiting patient based on modern criteria, evaluating them based on the modern criteria, then our actual approach will lose somewhere. So while designing the trial protocol, all these aspects has to be taken care. And as I said, that especially for non-pharmacological intervention like cupping therapy, or other dietotherapy, we have to take utmost care that how to design trial. Obviously, in these cases, it will not be randomized controlled trial, but maybe an observational study or some experimental study without randomization or without appropriate control. Then also we have to gather the evidence for to prove these efficacy of these traditional regimes. I'll not go to detail this. All you know that drug interaction, there might be chances of interaction of drug from her as well as food of her, because ultimately these plant material or minerals, they contain some phytochemicals which have effect on pharmacokinetic interaction sort of thing, and they may have some pharmacodynamic interaction sort of thing, and they can interact with the food as well as the drug which we are simultaneously taking. Because what happened, people will come up with our OPDs, they are already diabetic, so obviously they are taking some sort of metformin, and if they are hypertensive, they might be taking one modern medicine, and they don't tell to physicians actually that I am on some therapy, and they will take some our medicine as well. So that time it is a big risk to the patient. If we have any pharmacokinetic interaction data with those drug, that will be better. So initiation has been done by Ayurveda and our council to have data of pharmacokinetic as well as pharmacodynamic interaction of our commonly used formulation with, at least for commonly used medicine like diabetic for metformin, say for hypertensive drug, or some other long-term chronic disorder. So this aspect has to be considered while trialing design. So way forward is there is a need to implement stringent quality control measure and adoption of good agricultural practices, good manufacturing practices, and good clinical practices specified by ASU as well as CDSCO. There is a need to adopt flexible clinical trial methodologies to better mimic the holistic nature of traditional medicine while simultaneously ensuring the scientific rigor. Patient safety and well-being should be the utmost priority. Emerging technologies in various domains of drug development, including standardization, formulation development, and electronic data capture in clinical trial, must be adopted. Hub drug interaction studies must be conducted on commonly used traditional medicine, which are supposed to be co-administered with conventional drug. And there is a need of collaborative approach while designing clinical trial on traditional medicinal product involving experts from traditional and modern medicine. The development of standardized formulation with well-defined bioactive constituents followed by efficacy assessment through well-designed clinical trial is the need of time. So thank you for much. Thank you very much for patient listening. I would like to once again acknowledge my Director General and my Director in charge, Dr. Yunus Munshi Sahab. And I'm thankful to Professor Anil Kumar and our entire organizing team of IPS Conference 2025 for providing me this opportunity. Thank you.
Thank you, Dr. Gulam, for comprehensively explaining what are the challenges which we face when we are planning for pharmacological evaluation of traditional Unani medicine. So there are many aspects and multifaceted approach. We have to take care of these nuances when we plan for preclinical or clinical research. We do have various guidelines and regulatory provisions in place which will help you in designing these trials. Thank you so much. We have some quick questions. We are running short of time. We are getting requests from organizers to wind up this session as soon as possible. Otherwise, he's staying back, Dr. Gulam over here. You can discuss with him over a cup of tea
Sure.
or we can take one question. Anyone? So, thank you so much.
Thank you, Dr. Gulam.